Good morning, everyone. Right. All right, good morning. We will start with a new chapter today. Can everybody hear me okay? Hold on a second.
Yes, Dr. Bear, we can hear you.
I'm trying to tell. There we go. All right, I think my audio is better with the AirPods. All right, so today we are starting a new chapter. I'd like to remind everyone a couple of things about our exams. I posted our exam grades on Friday, and the average was a bit lower than I would have expected. I'd like to remind people about being prepared and ways to prepare for our exams. If you have any particular questions about your study approach or ways to think about this class, I welcome you to come to office hours. Today I have chat office hours at 12:30 in the web courses chat. I also have my Zoom office hours on Wednesdays, and you can always email me a question through our web courses inbox. You can, we could arrange for another Zoom if you can't come to my regularly scheduled Zoom hours. Remember, Joshua has Zoom office hours as well. But I'd like to reiterate some of the recommendations that I posted in the announcement with the exam. You who are here are coming to our lecture and taking in information in our regularly scheduled time. But remember, that's the beginning, right? That's the first step. I recommend that you watch the lecture video afterwards. Since we do record these in a regular face-to-face class, you wouldn't have a video to refer to. Use that to check your notes. Make sure that you have that you understand the topics that we've covered. Add the textbook readings to your notes and make some nice comprehensive notes. Make yourself a study guide and do all of that chapter by chapter so that you don't have 5 chapters worth of material to learn a week or a couple of days before the exam. And I recommend you doing this for all of your classes, trying to stay on top, learning, preparing for the next test all the time. Right. I know some people haven't been going to the lecture videos because several people asked me the day or two before the test how to access the Zoom. They didn't know how to, which told me that they hadn't tried before. Those are available for you to make sure that you're staying on top of our material. Right. And while you're preparing the study guides, that's that preparation, making those study guides gets you ready to show me what you know during the exam, right? If you try to rely on those notes during the exam, you won't have time to complete the test. All right, so if anyone has any specific questions on any particular question, or how you individually can prepare for success in this course for the next exam, come and see me in office hours or send me an email if it's a more general question, or if that's more convenient for you. All right. So today we're moving on to looking at our genome, and we're going to look at our genome in a slightly different way. We'll be looking at how the genome changes, how genes change, how new species arise, how genomes evolve over time. And in order to describe some of these changes, these heritable changes, how they occur, we first need to think about which cells those changes would arise in. And therefore, we need to distinguish between different groups of cells in a multicellular organism. We need to distinguish between our germline cells and our somatic cells. So our somatic cells are most of the cells in the body, the cells that make up that multicellular organism, the body, the soma means body, the cells of the intestine, of the muscular system, of the circulatory system. In contrast, we have our germline cells. These cells are actually set aside very early in development that are solely for the production of gametes. Right? Those cells set aside and will eventually undergo meiosis to generate the haploid cells that will carry that genetic information to the next generation. And so we want to think about a mutation that could occur in a somatic cell versus a mutation that occurs in a germline cell, what impact that has on that lineage, right? That lineage of organisms. And let's first of all take a look at our these cells in an organism, and then we'll look at the effect of mutations. So over here we have two generations in this of this species. The first, the parent organism, arises due to the fusion of two haploid germ cells fused to form a diploid zygote. Right? That single cell is the beginning of a new organism. That zygote undergoes multiple rounds of mitosis, giving rise to a huge number of cell types, and all of those somatic cells will differentiate over time and give rise to all of the different tissue types, right, to make up the organism. Some of those mitoses give rise to cells that become part of the germline, and those germline cells will eventually, upon reproductive stage, right, once this organism is an adult, will produce these haploid gametes. Right? And a haploid gamete will then fuse with another haploid gamete to give rise to the offspring, the zygote, which will then undergo multiple rounds of cell division. And again, we really want to focus on the gametes, right? The gametes, that sperm and egg, that haploid genome is what's carrying that genetic information to the next generation. So what if there is a mutation on one of these different cells of this parent? That's what we can see on our next slide. If we look at mutation B, a mutation that occurs in a somatic cell, that means that some subset of the cells in this multicellular parent organism may have a change. It may have a change in phenotype. But a mutation in a somatic cell is not going to be passed on to the next generation. Only a change in the DNA sequence of a germline cell may possibly be passed on as a haploid gamete to the next generation. And if that does happen, if one of the gametes that inherited this a genetic change in a germline, if that fused with another gamete to produce the zygote, then all of the cells, right, that arise from that zygote will carry that new change. And so this is what we're talking about in the ability of a genome to evolve in order for new species to arise, a change must occur in a germline, which will then be passed to the next generation of, of this organism. So what we want to spend our time here today is looking at what are some of these changes? What kinds of genetic changes will generate these heritable variations in genetic information? And we can break them down into several different classes of changes to genes, changes to the genome. And we've got these divided among two different slides. So our first group of changes that can give rise to alterations to genes that can allow a genome to change over time. The first one we're very familiar with. Here we have on the left-hand side, we have our gene, our transcription unit. And if there's a mutation within that gene, we end up with an altered product. Getting ahead of ourselves here. Meaning that a mutation, it could be a point mutation, it could be a deletion, it could be an insertion. If it's in the gene, it could result in the product changing, right? It could have a new function. Its activity could be modified. Its stability, how long it stays active in the cell, could be modified. It could have, let's say it's a protein that folds into a particular shape. It may interact with different other molecules in a different way. It may be localized to a different place in the cell. And then we contrast that to our second type of genetic change, if there is a change in a regulatory region, right? The region to which is going to determine when and how much and in which cells that gene will be transcribed. So if we have a mutation in a regulatory region, maybe an enhancer, maybe a suppressor, maybe within the promoter region itself, we can end up with altered expression. That gene product may be produced, but it may be produced at the wrong time in the life cycle. It may be produced in the wrong cells, or it may not be produced in a cell that requires that gene product. Right? So here, this is how we're distinguishing mutation within the gene itself, encoding the function of that gene, and mutation in what's regulating the how and where and how much and what time that gene is going to be expressed. And then we can contrast that to a different type of genetic change that we call a gene duplication, where a gene can be duplicated, that genetic information can be duplicated to form a gene family. And a gene family produces a set of related gene products, a set of related proteins. These proteins are related because they are encoded by a very similar nucleotide sequence. But because there's two copies of this gene, it's possible for each of those copies to accumulate different mutations, secondary mutations, over time. And they may take on slightly different versions of the function of the original gene. And we'll look at examples of that today. They may do, and when I say they may accumulate different mutations, they may diverge. So a gene family first arises by the duplication and then the subsequent divergence of those two versions of the gene. And then we can move on to another group. Here we're just looking at them in general, and we'll look at specific examples a little bit later. Sometimes we can have new forms of genes arise. Up here we see our orange gene A consists of 2 exons. Red gene B consists of 2 exons, but different types of genetic processes like unequal recombination, transpositions can exchange some genetic information so that we end up with new versions of gene A. Gene A may pick up an exon from gene B, and it may exchange its exon for another exon of gene B. So we can rearrange these genes to form new versions and therefore products that have different functions. It's also possible, and this can be driven by mobile genetic element movement, number 4. We'll see an example of that later, but also mobile genetic elements, just by virtue of them moving, they may jump into a regulatory region. And so the transposition of a mobile genetic element may affect a gene. It may affect the expression of a gene. It may change where or when or how much that gene is expressed. We'll see examples of that as well. And then we have our 6th type of genetic change that we can consider horizontal transfer. That is transfer of genetic information from one cell to another cell. It could be another individual, it could be another species. And this species or organism B has now picked up, it has acquired new genetic information due to this horizontal transfer. And we'll look at a couple of examples of that later as well. So what are some of these changes? Now, our very first one, the first example of a mutation within a gene, right? A mutation that affects, let's say, the coding region of a gene. We know about that. We've talked about that in genetics. We've talked about that in our previous chapter as well. The mutation in a coding region or mutation within the transcribed region of a gene can result in an altered product, right? The product either maybe has no function, or the product has a slightly different function, or it's not as stable in the cell. Those kinds of changes are acquired by altering the transcribed region of the gene. But many changes occur in regulatory regions, not the region of the gene that's transcribed, but the region of the gene that is regulating where and when and how much is expressed. And the example that we can look at here is, on our next slide, is the effect of a mutation or a change in a regulatory region can give rise to differences in organisms that, while the change at the DNA level can seem to be very subtle, can create major changes in the body plan and in the development of organisms. And this really is where most changes are occurring during evolution, changes in genes that are very early, that have effects very early in development. So here we're looking at 2 mammals. There's a lot of similarities in these 2 organisms, but there are some distinct differences in morphology, of course. And if we look at organism A and B, we see that they carry, in this region of the genome, three genes that are the same. They both carry gene 1, 2, and 3. And if we look at very early in embryogenesis, both organism A and organism B produce this gene 1, the product from gene 1. And the product of gene 1 is a transcription factor, which means that it will bind to other genes and activate their transcription. So they both do the same thing. Both these, our little anteater on the left and our dog on the right, produce this transcription factor, our brown triangles early in development. Later in embryogenesis, what we see is that organism A, that transcription factor, binds to a regulatory region in front of gene 3. And by binding, right, it can recognize the DNA sequence, will bind to it and will activate transcription of gene 3, which produces these blue triangles. And let's say this is another transcription factor that will then activate another set of genes, right? And essentially activate a transcriptional pathway that's going to lead to development of this organism. And the difference between organism A and organism B could be as simple as the transcription factor that's encoded by gene 1 will, in organism B, bind to a regulatory region that's in front of gene 2 instead of gene 3. So the difference between these two species is the location of a binding site for our brown transcription factor. Right? In organism A, that DNA sequence is in front of 3. It produces one type of transcription factor that regulates other genes. And in organism B, that same sequence is in front of gene 2 instead of gene 3. And so a green square transcription factor is activated. So relatively subtle differences in regulatory sequences in front of genes is going to dictate the expression of different genes that are going to have big impacts on the development of these related but different organisms. So I'd like you to think for our first clicker question today, what are other possible changes that could occur in regulatory regions of genes, and what could the effects of those changes be? And that's what I'm going to ask you to identify here. So in this question, I'm asking you, of these 4 different changes, different mutations that occur in a genome, which one is least likely to arise from a mutation in a regulatory region? Right? So think about, what do regulatory regions of genes do? Right? They're going to dictate when in the life cycle, when in development a gene is expressed, the when, the where, the which cell types that gene is expressed in, and how much that gene is going to be transcribed. So which of these would be least likely, or which of these changes would not be due to a change in regulatory region? How are we doing? How about 5 more seconds? All right, let me close it up. Okay, the one that was least likely. Now I see that a lot of people have chosen B, but I'd like you to consider why C is the best answer here. So C is the correct answer, the one that is least likely. And let's look at why. Why is C the correct answer? So if there's a mutation that changes the time, right? That would be a mutation that would be very likely. That's a mutation in the regulatory region, right? It's changing the timing of that gene's expression. B could be a mutation in a regulatory region that is going to eliminate transcription of that gene product, right? And therefore, if the gene's not transcribed, it can't be synthesized. It can't synthesize a protein. D, a mutation that increases the level, that's going to be a mutation that is changing the amount of the gene product, right? So A is when, B is where, right? Which tissue type, and C is how much. If you have a mutation that changes subcellular localization of a protein, when we say subcellular, we mean where, right? Where in the cell. Subcellular localization, for example, if there's a mutation in the nuclear localization signal, or a signal that sends it to the mitochondria, or a signal in the protein that sends it to the ER, right? Those would all be mutations in the protein coding region. It's going to change the 3-dimensional folding of that protein, right? Because a protein is going to be localized based on the amino acids on the surface. What it interacts with is going to determine where it will function in the cell. Okay, so yes, you could say that you could have a mutation in a protein coding region of a gene that would knock out that gene, right? Prevent it from being translated, but it wouldn't prevent its translation in just one cell type, right? It would prevent its translation anywhere. Right? So C would be due to a mutation in the coding region. All of these others could arise from a mutation in a regulatory region. Okay, so I'd like you to think about that. Regulatory regions of genes are determining when, where, as in which cell types, and how much that gene is expressed. So let's move on to our third type of change in genetic information, change in a genome, something we haven't really talked much about before in this class, or even in genetics either. And this is gene duplication. And this is very common in organisms. And as a matter of fact, a large portion or large proportion of vertebrate genes are members of gene families. And what that means is that we have a group of genes that originated by that gene being duplicated, sometimes more than once, and then different members of the gene family diverge. Different members accumulate different mutations that give them slightly different functions in the cell that could be selected for if there's an advantage. And so we're going to take a look at the globin gene family. Globins are polypeptides that make up the hemoglobin that carries oxygen through our blood. There are different members of the globin gene family that are expressed at different times of development, in fetal development, embryonic development, as well as in adults. And we also see there's some pseudogenes in that gene family as well. So let's look at an adult version of our hemoglobin, the hemoglobin that we carry in our red blood cells. It originated as an ancient single chain globin polypeptide. And this actually is still present in some very primitive vertebrates as well as insects, carry oxygen bound to this heme group that is bound to a single globin molecule. Over the course of evolution, this ancient globin was duplicated. And the duplication of the gene that codes for a globin allowed for more mutations to arise. So we had to produce a slight variant on the original globin, such that we now have a hemoglobin protein that consists of 2 beta subunits, meaning 2 beta globin polypeptides, and 2 alpha globin polypeptides, each of which has bound to a heme molecule, and each heme binds an O2 molecular oxygen. And this is a great advantage for carrying oxygen through the blood, not only because this molecule carries more oxygen, but also because there's a relationship, an allosteric relationship between all of those subunits. As soon as one subunit binds a molecule of O2, it changes its shape slightly, which then makes it easier or more efficient for the other 3 subunits to bind oxygen. Right? So working together, they carry oxygen better than 4 times 1 globin molecule. So let's take a look at this gene family. Our gene family here is, we're looking at 2 different essentially regions of our globin gene family that are carried on 2 different chromosomes. On chromosome 16, we have what we call the alpha globin locus. On chromosome 11, we have the beta globin locus. And our alpha locus consists of multiple members of the gene family. One, this zeta that's expressed in embryos, 2 versions of the alpha globin that are expressed in both in fetus and adults, and our beta globin locus has two gammas expressed in the fetus, as well as a beta globin and a delta globin expressed in adults. Different globin proteins have different affinities for oxygen, meaning different versions of the globin polypeptide fold into a shape such that they bind oxygen in slightly different ways. Well, we can also see that at both of these loci, there are some pseudogenes. Now remember, a pseudogene is essentially an evolutionary relic. This is a copy. These pseudogenes are copies of these other globin genes, but they have accumulated mutations that make them non-functional. So we can see the sequence, the DNA sequence that identifies them as having in the past been functional globin genes, but over time, they're either not transcribed or they're not translated anymore. So you might ask then, why do we have multiple globin proteins? Why is there a form that is more common in the fetus? And why do we have, for example, at the globin level that are commonly expressed, differentially expressed in the adult? And the reason is that when the fetus is developing inside the uterus, that fetus has to be able to compete for oxygen in the mother's blood. And these fetal versions of hemoglobin, both from the beta globin locus as well as from the alpha globin locus, bind oxygen more efficiently than the adult forms do. And that means that the developing fetus can compete better for the oxygen in the mother's blood than the mother's hemoglobin. Then after the fetus is born and is air breathing, then it transitions eventually to the adult forms, which are slightly less efficient in binding oxygen, which means that they'll be able to release oxygen a little more efficiently, which is better for an air breathing organism. So over time, over millions of years, these different forms of these globins have evolved that are expressed differentially at different stages in our life cycle. So how did these arise? How did these different variations, these different versions, different members of the globin gene family arise? Again, we started with a single chain globin, and somewhere about 500 million years ago, there was the first big event. The first big event was that there was a duplication of that single chain globin, giving rise to an alpha globin and a beta globin. A second big event resulted from the breakage of the chromosome that separated the alpha globin genes from the beta globin gene. The alpha globin gene ended up on the chromosome that we now call chromosome 16. The beta globin gene ended up on the chromosome that we now call chromosome 11. About 200 million years ago, the beta globin gene duplicated again, giving rise to a fetal form and an adult form. And then each of those, as we can see, duplicated again. Once one duplication occurs, we'll see a little bit later, it's very easy, relatively easy, for other subsequent duplications to arise. So we have multiple events that have occurred, giving rise to these multiple members of the globin gene family. And here we're only looking at the beta globin locus, the same kind of thing, multiple duplications and subsequent divergence, right? Subsequent accumulation of new mutations occurred at the alpha globin locus as well. So based on that evolutionary history that we just looked at, which of these following statements is true? Oh, sorry. There you go. I'm seeing lots of problems. Was there a problem with the last question? Did I select the wrong answer? Someone's, I'm seeing questions about where. Remember, there's where as in which cell types, and there's where as where in the cell, right? Those are two different types of wheres. So which of these statements is describing this evolutionary history correctly? Everybody had a chance to vote? How about 5 more seconds? All right, let me close it up. All right, and the one statement that is true is D. So what's wrong with the first one? Ancestral globin was present more than 500 million years ago, and it was that first duplication to give rise to alpha and beta that occurred 500 million years ago. The alpha globin is equally distant to both the epsilon and the delta globin because both the epsilon and delta globin share the same common ancestor. So alpha globin is not more closely related to one or the other. And C is not correct because even though, yes, the two gamma globins are closest to each other, but that doesn't necessarily mean that they diverged from each other most recently. Just because they're closest, it's actually looking at the sequence of these different genes allows us to determine which are more closely related and which diverged from each other most recently. We'd have to look at the sequence to make that determination. And so the only one that's correct describes the gene duplication event that occurred about 200 million years ago that gave rise to beta globins expressed in fetus that's different from the beta globin that's expressed in the adult. And those different types of expression patterns would be due to accumulating mutation in regulatory region, right? They're regulated in different ways, as well as mutations in the coding region that gives rise to slightly different structure to each of those different types of globins that changes their function, how they bind oxygen. So how did these duplication events arise? The most common way is by what we call unequal crossing over. And the unequal crossing over arises because, as we know, our genome is sprinkled with these repeated sequences all over the place. These short repeated sequences, like signs, like the ALU sequences in humans, are scattered, but they may be present in the same gene. Just maybe by chance, there may be multiple copies of a repeated sequence in a particular genetic region. And that can give rise to unequal crossing over events. So let's take a look at one region of our genome. Here we have a pair of homologous chromosomes that have the same genetic information, right, in one parent. And if this cell is about to undergo meiosis, then we have synapsis will occur at the beginning of meiosis, where the homologous chromosomes will line up, right? They line up gene by gene based on the similarity of sequences, right? And that synapsis will then allow for, at the very beginning of prophase, the possibility of a crossover event, right? It could occur anywhere along those homologous chromosomes. It may occur within the gene or next to the gene, and we may have exchange of genetic information between our top chromosome and our bottom chromosome, right? The non-sister chromatids between of those two homologs. And what we would see is that their genetic information would give rise to, like I said, exchange of genetic information. But if we have these short DNA sequences, repeated sequences near that gene, during synapsis, the two homologous chromosomes may misalign. They may line up based on the similarity of these repeated sequences shown in red, rather than the gene, the transcribed region itself. And that misalignment, when that recombination event resolves, could result in exchange of genetic information that gives us what we call an unequal crossover event, where one chromosome actually picks up, there's our first copy, crosses over and picks up a second copy of the same gene. Now we have a duplicated copy, and the other chromosome has lost that gene, right? It's lost a copy of the gene. Once we have a duplicated chromosome, we could see that in subsequent generations, it would be very easy. Now we have more regions that are in common within this short region of the genome. One duplication event makes it very easy for other non-homologous recombinations to occur, which is one reason why it's very common for gene families to contain more than 2 copies, but could have 4 or 5 or 6 or more. And again, in vertebrates, we have many of our genes are members of gene families. Our G proteins are members of gene families. Immunoglobulins are members of gene families. The Hox genes that are transcription factors in early development. Motor proteins like myosins, different types of myosins that have different types of functions. Those motor proteins are all members of gene families as well. Now, it's also possible, again, these are sort of accidents that occur during cell division that can give rise to alterations that if there's an advantage to the individual, right, will be maintained in subsequent generations. But it's even possible for an entire genome to be duplicated. And you can imagine that occurring if the germ cell, instead of undergoing 2 rounds of cell division, or if the cell fails to divide after the DNA replication, the cell may end up with a doubling of its genetic information, a doubling of the genome. And that could possibly be passed on to the gametes and then eventually to progeny. And this type of duplication actually does occur and has given rise, it's relatively common in amphibians, and it's given rise to about 20 different species in the Xenopus genus. If we look over here, just as one example, up here we have the diploid version of this Xenopus species. Xenopus tropicalis has 2 copies of all of its chromosomes, just like we do. But a related species, Xenopus laevius, has resulted from the duplication of its genome, and so now it is polyploid. All of its cells contained 4 copies of all of the chromosomes. And all of those different Xenopus genuses have multiples of a diploid genome. They can be 4N, they could be 8N, they can be 12N, they can be 10N, giving rise to related but distinct species of this type of frog. So moving on, we're going to look at different other ways, right? We're just very briefly looking at different things that can happen to a genome that can give rise to variations that may be selected for during the course of evolution. And the next 2 examples involve our mobile genetic elements again. When they move, right, transposition, when a mobile genetic element moves, it might actually cause a rearrangement of some genes. And we're going to look at one example of that on the next slide that we call exon transposition, the movement of an exon of a gene that gives rise to a novel gene. So what we have up here is we're looking at a locus in blue of gene A that consists of 3 exons, exon 1, exon 2, and exon 3. And in the large introns of this gene sit 2 different copies of one mobile genetic element. And sitting in the introns, it's not really a problem because, of course, when this gene is transcribed, the introns are spliced out, right, joining exon 1 and 2 and 3 to produce a mature RNA that can be translated. But the problem arises if these mobile genetic elements mobilize, if they transpose, if they move away. Now, in some cases, the element will cleave itself at its ends, those inverse repeats at the ends that are recognized by the transposase, and sort of pop that mobile genetic element out of the intron. Right? The cell will never know the difference. But in other cases, again, having multiple copies in the same region of the genome, the transposase may cut the end of one mobile genetic element and cut the end of another mobile genetic element and end up popping out the wrong segment, pop out a region of that gene that includes exon 2. And mobilization means that it moves from one place to another. And it's possible that that mobilized element may pop itself into gene B. It may pop into the intron of gene B. Now, gene B in green consists of 3 exons, again, separated by large introns. And this removed element from gene A may pop into the intron of gene B, producing a new novel arrangement of exons. Now we have a new version of gene B that consists of 4 exons. Now, again, we would have to assume that this novel gene product would arise only if, right, the splicing from exon 2B, splicing to exon 2A, and 2A to 3B would remain in frame, right? But if that does happen, it may actually add a new functional domain to the protein that's produced by gene B. But this is one example of what we call exon transposition, where a mobile genetic element has rearranged a gene to form a novel version of that gene that, if it enhances the ability of the cell to survive and reproduce, may be selected for and may then create a new form of that gene, a new form of that gene product. Now, these transposable genetic elements, again, can jump into the transcribed region of a gene, but they can also jump into regulatory region of a gene. And when they jump into the regulatory region of a gene, they can cause some changes to that gene's expression. Right? Won't change the product, but won't change the function of the product, but can change where and when that gene is expressed. And an example of that we can see on this slide. This is very well understood in genetically amenable organisms like Drosophila, where there are some early embryonic transcription factors that are going to determine the body plan, how this organism is going to develop, how it's divided into the segments: head, thorax, abdominal segments. And Tenopedia is the name of one of these transcription factors that's required for the formation of legs, right? 3 pairs of legs as this animal is developing. However, there are some mobile genetic elements that over time have moved around within the Drosophila genome, and they're called P elements. One P element that has jumped into the regulatory sequence of Antennapedia has changed the expression pattern of Antennapedia. And in this mutant form, Antennapedia is expressed not only in cells that should give rise to 3 pairs of legs, but Antennapedia is expressed in the head region. And we call that ectopic expression. It's expressed in a place it's not supposed to be expressed in. And when it's expressed in the head, Antennapedia will drive expression of genes that give rise to legs, but the legs are being formed in the head. We call this a homeotic transformation. Instead of antenna forming in the head, legs will form there. This kind of, now this is an extreme version, of course, but this kind of mobile element change in gene expression patterns has been very important to evolution of species, changes in body plans of organisms over time, both plants and animals. And if we look on the next slide, we're just looking at a little bit more detail at this type of homeotic transformation when one body part is replaced by another body part. Instead of, on the top left here, instead of normal antenna and aristae sensory organs being produced or formed in the head region, a pair of legs are there instead, due to a mutation in a regulatory gene caused by a mobile genetic element jumping into that regulatory region, causing that gene product to be expressed in the wrong place at the wrong time. Now, our last example, and let me make sure we're not running out of time here, 1 more minute. Our last example is genetic changes that contribute to gene alteration. And this we consider to be horizontal transfer as opposed to vertical transfer, which would be from parent to offspring. Horizontal transfer is movement of genetic information like this red gene from one organism to another. Could be from one individual to another in the same species, or could be from species to species. And a couple of examples of how that can happen is this actually occurs very frequently in bacteria. And that process we call conjugation, where DNA carried on a plasmid is transferred from one bacterium to another. For example, this genetic transformation from this one cell here at the top will form a sex pilus that will allow for transfer of plasmid, and which may be carrying antibiotic resistance genes, transferring that genetic information to another cell, another bacterium. Again, very common. E. coli, the bacteria that colonizes our gut, has picked up over the course of its evolution, about 20% of its genome was picked up by conjugation from other species of bacteria. And not only transfer of DNA from one bacterium to another, but viral infections can pick up genetic information from one host when that virus forms its new particles and is released and then infects another host. It may actually pick up some genetic information from the first host and transfer that to a second one. Another example of this type of evolution, genetic changes, in this case, it's horizontal transfer. So I am sorry, we have run out of time. I have one more question about this. I'll bring this on Wednesday. We will start our next chapter, but I've got a couple more questions to ask you about this chapter, chapter 9, on changes leading to genome evolution. All right, so we will start a new chapter on Wednesday. I have office hour chat at 12:30 today, and you are free to send me any email questions if you have any specific questions on material, study habits, or even the test this week as well. All right, see you on Wednesday.
Thank you. Dr. Bear, is
You're
there like
welcome.
a section for like what we need to know for this chapter?
I do, but I don't have it here yet, so I'll
Okay.
give it to you. I'll put
Okay,
it up.
perfect.
Okay,
Thank
I
you.
ran out of time. Okay, bye-bye.